Right, so we are now being recorded. So this is our 11th and second to the last video teleconference in BIOL 105 YA. Um, I will not go to the presentation mode yet because some students might be asking me to for permission to join the video teleconference, no, and I experienced some lags yesterday when I'm exiting and going into presentation mode. So as for the meantime, up to around 8:15, I will be in this non-presentation mode first. So I hope you can see my slides. So this is our calendar for April. We are now on the 20th and we are doing our 11th video teleconference. So ahead of what's scheduled here. Um, And our topic this morning is actually a discussion on cell signaling. So I did not post any lessons on this. So the recording of this will also serve as my posted video later on. Um, so next week, we still have to cover our last chapter, no, chapter 17, that's the immune system. And our video teleconference here is highlighted in yellow on the 30th of April. So this will be our last video teleconference. So after this, we are done with our course. So you just need to wait for the final exams, which will be around second week of May. It's the second week of May. So are there any questions or concerns regarding our schedule? Wala ra. Okay, so we're almost done with our semester, no? So I hope you are also wrapping up with all of your requirements, not just for this course, but for the other courses as well. So without further ado, let's start now with our topic this morning, cell signaling. So cell communicating with other cells or with itself. Okay, so in this first slide, okay, we can see here on top, no, on top of this broken blue line, cells that are communicating to the cell beside them, no, the adjacent cells. So here on the illustration on the upper left, you can see two types of cells, animal cells and plant cells, communicating with their adjacent cells via cell junctions. And I think you can remember this, the gap junctions for animal cells provide as passageways for chemical signals to move from one cell cytoplasm to the other cells. While in plant cells, the equivalent of that is the plasmodesmata. So this is very self-explanatory, know how chemicals move from one cell to another adjacent cell. Well, here on the upper right, we can see two adjacent cells again, but this time they are communicating not via junctions, but via the molecules on their cell membrane surfaces. So you can see here a receptor that's colored purple on one cell receives a signal from a molecule that's colored green on the other cell. Okay. And I think we can recall in the previous discussions that this could be carbohydrates, no? Uh, that are interacting with each other, allowing cells to recognize each other. So these are ways of communicating between cells. But our topic this morning um, is also cell communication, but we will focus on the release of chemical signals that cells later on receive. No, so what am I trying to say? This is different from the communication na cell-to-cell junction communication or surface glycoprotein communication. We are talking here right now about chemicals being released by the cell so that it can be received by another cell later on. Okay. So, what are examples of these? It can be the cell releasing a chemical, and the recipient of the chemical signal is the cell itself. No, so you can think of this as you writing a reminder for yourself. No, have you ever done that one? When you need to remind yourself on a task that you need to do later in the day, so you won't forget it. You write it in a notebook or in a sticky note, and then you read it later to remind yourself. So that's what we see here, no, at the bottom, kaning pinakauna, the cell reminding itself or communicating to itself. Next, the cell can also release chemical signals to communicate to the cells beside it. So this is like during the face-to-face classes, no, when you talk to your seatmate, you're communicating directly, no, through exchanges of words, no, when you converse. So this is what we call paracrine signaling. Paracrine signaling. So going back to the cell communicating to itself, this is called autocrine. Autocrine signaling. Well, cells can also communicate to other cells that are far away from it, just like what we're doing right now. No, we are very far from each other, but we are able to communicate because of this internet. And that's what we see here on the middle. One cell releases a signal, it goes into the blood vessel, no, and the signal flows in the blood and then reaches the destination and reaches the cell, the recipient, no, and the message is read by the recipient cell. So it's like the blood vessel is the internet connection. Now, and my signal right now are the words that I'm speaking, and then you're hearing it there in your end because of the internet connection. That is what we call in cell communication as endocrine signaling. And we have the endocrine system, if you can remember, which uses hormones, no, as signaling molecules to give signals to cells that are away from the signaling cell. And lastly, here on the bottom right, we can see neuronal signaling. So this is a neuron, a nerve cell, releasing neurotransmitters to another cell via the synapse. Now, we can also call this a synaptic signaling. Now, for this is largely involved in the nervous system, for example. So I will now go to presentation mode. Okay, can you still view my slide?
Yes, sir.
All right. So I hope this is clear to you. Know, these are the different ways by which cells communicate to each other. So our focus is this chemical signaling. Kaning nasa ubos na mao na tong focus. Particularly the first three here. No, wala na ng neuronal signaling because we have talked about that a bit in the previous lessons. Okay, so now let's look at the details. Here we have the signaling cell releasing a signaling molecule. We also call as the first messenger. And the recipient cell, the one here at the bottom, has receptors on its membrane. And the signaling molecule can actually bond to the receptor like a lock and a key. No? So in that sense, we call this signaling molecule as a ligand. Ligand. No? So I hope you're familiar with the term ligand. Let me admit someone in first. Okay. So we have the signaling molecule, which is a ligand released by the signaling cell, which binds to the receptor of the recipient cell. Okay. And then what we see in this figure are two possibilities. Here on the left, the receptor activates an effector. Now, there's an effector, and the effector makes a second messenger. So, naapag yung second messenger. So, gipasa niya ang message sa second messenger, and the second messenger will be activating other proteins along the way, as we see here at the bottom. So um, on the right is another possibility wherein the receptor, as you can see here, there's a letter P here. It phosphorylates a protein, and that phosphorylated protein becomes activated and activates other signaling proteins along the way. And both of them, as shown here, results to the reaction of the cell, no, as we see here at the bottom. Okay, so the receiving of the signal is called signal reception, and the passing of signals to different molecules, no, is called signal transduction. So these are important terms, no, that you should be familiar with. There is reception, and then there is passing of the message through different molecules called signal transduction. Now I have here an illustration on the right. What's the difference? No, can anyone in the group tell what's the difference between this illustration and the two others here on the left? What's happening to the signaling molecule? Is there a receptor involved? Is there a receptor here on the membrane, as you can see? Yes or no?
No, sir.
There's none. But the signaling molecule, look what happens to it. It goes directly into the membrane. And the receptor is inside the cell, not on the membrane, but inside. No, so that's the difference here. Sa these first two examples, ang signaling molecule stuck to the receptors on the surface of the membrane. But here on the right, it went directly inside the cell. So what's the difference there? Um, can you say that this signaling molecule is hydrophobic or hydrophilic? Will we be able to tell if it's hydrophobic or hydrophilic? What is your guess? Because it has to pass through the hydrophobic cell membrane man. So can a hydrophobic or hydrophilic molecule pass through? So what should this be, hydrophobic or hydrophilic?
Hydrophobic, sir.
Yes, no. So if you can remember your chemistry, like dissolves like. So this has to be hydrophobic in order for it to be able to pass through the hydrophobic cell membrane. So an example of this na, if you notice, no, kanang if you have rashes or naikatol-katol, the ointment that you put on your skin, di ba, it's oily. Okay? The reason why it's oily is because that actually allows it to easily penetrate the membranes of the cell on your skin, for example. So, dali lang siya ma-absorb sa imong skin cells if it's hydrophobic or oily. So, that's what's shown here. Okay. So, let's go to the detail of that signal transduction. No? So, kaning sa ubos, how does one molecule pass the signal to the next? The answer is it actually activates the next molecule. So, look at this. First, you have an inactive protein kinase. I hope you can remember what a kinase is. What is a kinase? Anyone? Kanang term nga kinase, what type of enzyme is that?
It adds phosphate groups to other proteins.
That is correct. Now, so in signal transduction, there are kinases involved. So look at this. So I have protein kinase 1, which will activate protein kinase 2 by adding phosphate. to it, no, phosphorylation. And then this one, protein kinase 2 na active, will activate the third one by, again, phosphorylating, and so on. So this is how signal transduction happens, no? Phosphorylation of different kinases. Okay, so this is just a detail, no? The phosphorylation usually happens in the amino acid residues na tyrosine, serine, and threonine. So particularly kaning tyrosine, we will be talking about this term again later on. So please don't forget about tyrosine na naay phosphorylation. So overall, the beginning of cell signaling is because of the extracellular messengers, which include amino acid derivatives, nitric oxide and carbon monoxide, steroids, icosanoids, binding to the receptors like GPCR. Say meaning sa GPCR. Katong mga nakabasa na. R here stands for receptor. So this is
G protein, sir.
all right, G protein coupled. So meaning to say ang receptor is coupled with the G protein. G protein coupled receptors. Then you have RPTKs, no, as also receptors. Receptor protein tyrosine kinases. So tyrosine. So remember the word tyrosine again. Okay. And then we have ligand-gated channels, and then steroid hormone receptors and antigen binding receptors. So particularly in this lesson, we will focus in the first two, kaning GPCRs and RPTKs. So the extracellular messenger, which is a ligand, will stick to the receptor, and that's when the signaling begins. So let's go to the first type of receptor, the G protein coupled receptor. Okay, so this is the receptor, kaning green, no, that you see here. It's a transmembrane protein. And on the inside of the membrane, we have this protein subunits composed of alpha, beta, and gamma. This is the G protein. The reason why it's called G protein because it's bound to guanosine diphosphate. Okay, when a ligand or a signaling molecule sticks to our GPCR, this green one, it will cause the sticking of the G protein to itself. So now, nadaday contact between them. And once this happens, this will facilitate the exchange of GDP for a GTP. So nahimo na guanosine triphosphate instead of guanosine diphosphate. And once this happens, this alpha subunit of your G protein will be activated and it will go to an effector protein, as we see here. So look at this. The alpha subunit will go to the effector. This will cause the activation of the effector. And the effector, in this case, will make cyclic adenosine monophosphate. So this is an example of a second messenger. No? Ang first messenger na to, kaning red, tong signaling molecule. And then this is now the second messenger, the cyclic adenosine monophosphate created by the effector protein, which was activated by the alpha subunit of the G protein. Okay, so take note of the sequence. No? This is very basic in cell signaling. And once the adenosine monophosphate is made, the GTP on the alpha subunit will be hydrolyzed to become GDP, and the alpha subunit becomes inactive, and it will return back to the other subunits. So the cycle will be able to repeat itself. No, the signaling will be able to repeat itself. So that's the sequence. No, so unsa man ang ultimate goal ani? The making of a secondary messenger or a second messenger. Okay. So are there any questions or concerns about this? Important kayong i-remember ang effector protein, no? Okay. So the question now is this. Okay, we're done with the signaling, no? Nahitabo na. How does the cell stop that? No, because just like us, the cell doesn't want to be told again and again what to do. No, so for example, ang signal dire kaningon, okay, cell make some proteins. So once the cell receives the signal, the cell doesn't need that instruction to be repeated again and again. So it has to be stopped. There has to be a termination. Okay? And that termination, as we see here, is begun by the binding of a protein called GRK. Okay? G protein coupled receptor kinase will bind to this GPCR, and the binding causes the phosphorylation which attracts arrestin. So na ay molecule ng tawag arrestin. Now, this will stop the binding of the G protein to your receptor. And so there's a termination of the response. What's more is that this G protein coupled receptor that's bound to the arrestin is endocytosed. Look at this. There is this clathrin coated endosome. So this will be endocytosed, and the cell can either decide to destroy the receptor in the lysosome or recycle it back to the surface later on when needed. So this is how the G protein coupled receptor signaling is terminated. So any questions on this? None? Okay, so let's continue. Let's have some examples of the second messengers. No, a while ago we are already acquainted with CAMP, or cyclic adenosine monophosphate. Okay, this is how it looks like. It's like ATP with only one phosphate, and then there's this cyclic shape over here. So the question is, what is this effector molecule? The detail of this, the one that makes cyclic adenosine monophosphate, is this. It looks like this. Now, and this is an enzyme known as adenylyl cyclase. So the effector protein here pala has a name. It's called adenylyl cyclase, and it's the one that catalyzes the formation of cyclic adenosine monophosphate from ATP. So this one will become the second messenger. So another thing to remember. And then another second messenger would be the inositol triphosphate and the diacylglycerol. So what are these? If you can remember your membrane lipids, cell membrane lipids, one of them is this phosphatidyl inositol. So from here, this can actually become your IP3 and your DAG. So this is how it looks like. No, we have the tail and then we have the head. So there is an enzyme called phospholipase C, or PIPLC, which actually cuts putlo niya imong phosphatidyl inositol into two parts. The one on top is your inositol triphosphate, so this one, and the one at the bottom is your diacylglycerol. Okay, so these two, once cut, become second messengers. Okay, so gikan sila sa kaning phosphatidyl inositol nga phospholipid. So how does that happen? So here we have this kaning teal nga layers mao ning cell membrane. And we have here illustrated, this is your phosphatidyl inositol. Okay, so let's look at the role of phosphatidyl inositol in the release of calcium ions in the cytoplasm. So first, it has to be phosphorylated first, kaning head portion with a kinase. So look at this. Kinases will phosphorylate the head first. Okay? Once that's done, um, uh, the activation of G protein, as we see here, so G protein coupled receptor gihapon no, na kay ligand, na kay G protein coupled receptor, which will activate the alpha subunit of the G protein. And this time, instead of going to your adenylyl cyclase, the effector in this scenario is your phospholipase C. And phospholipase C will cut your inositol, phosphatidyl inositol into diacylglycerol, and the head is going to be this one, the inositol triphosphate, which will go inside the cell, which will bind to the inositol triphosphate receptor in the smooth ER, which will cause the release of calcium ions from inside the smooth ER into the cytoplasm. So look at this pathway, this signaling pathway. So this is another example of the G protein coupled receptor signaling pathway. Okay, so are there any questions so far? How is this the same as the one with the adenylyl cyclase? Anyone? Katong adenylyl cyclase ganina. I may go back to that slide. Ani, how is this different from this one? The same ba ang ilang receptor sa katong adenylyl cyclase? Yes or no?
No, sir.
Pareha, oy, G protein coupled receptor. Tanawan ning R ha. Balik ko dito. Awa, kani. But this is the receptor, kaning green, no? Okay. That's the G protein coupled receptor. They are the same. Okay. So, pareha sila. So, take note of that. What about in the G protein, kaning alpha, beta, o gamma? Pareha, dili. Kato ganina, na ba to'y alpha, beta, o gamma? Wala.
Yes, sir.
Oh, naagi hapon. So, pareha. So, take note of pareha sila. Okay? But, when the alpha subunit is activated, ganina, man to padulong ganina. Itong in the previous slide.
Yadto sa effector.
Which is, sa tungalan sa effector?
Adenylyl cyclase.
Okay, an adenylyl cyclase will make what?
Cam.
Okay, which is a second messenger. Now here, in this example, pareha sila sinugdan, pero the alpha subunit this time goes to a different effector, which is phospholipase C. And the resulting second messenger is not CAMP, it's this. It's the inositol triphosphate. So see, pareha ra ang ilang receptor o ang G protein, pero ang effector, diha sila naglahi. Maupod niya ang iyang resulta, lahi nga second messenger. Okay, so it's important for us to be able to see that no in the illustrations. So here again, the second messenger is inositol triphosphate, and it causes the release of calcium ions from your smooth ER. Okay, so mao na siya no. So please be wary of the illustrations. So narang iyang mga functions, no? Smooth muscle contraction. So remember your calcium ions and katong tropomyosin and troponin, di ba? So ang muscle contraction is also facilitated by calcium ion release, di ba? Okay. And we also have here the formation of fertilization membranes in eggs. Okay, next. Umanata atong kuan ha, inositol triphosphate. So remember that. Karon, let's look at the camp. Let's go back to the camp. Mura taga camp no. So camp, cyclic adenosine monophosphate. Okay. Now this time, let's look at its role in glycogen metabolism. So here, the same story. You have your G protein coupled receptor. You have your G protein, parihara ng astorya. But this time, the alpha subunit goes to adenylyl cyclase. So ang yang himoon, once active, adenylyl cyclase will make cyclic adenosine monophosphate. So ganina lahi, instead of adenylyl cyclase, that was phospholipase C. Karon, adenylyl cyclase. So your cyclic adenosine monophosphate will cause the activation of protein kinase A. Ma-activate si protein kinase A. And look at the result here. When active, protein kinase A will inactivate your glycogen synthase. Unsay meaning anang glycogen synthase? Siya ang tighimo og unsay ganahimo sa glycogen synthase?
Ang glycogen.
Glycogen. So, the result here is it will inactivate, stop niya si glycogen synthase. Now, in order to understand this, let's look at the ligand here on top. Unsay ngalan ani nga ligand? Glucagon. Do you remember glucagon? No, the opposite of insulin. No, in sort of the way it works. And here we also have epinephrine. No, the hormone that will allow you to burn your energy so that you can respond really fast. So look at the result. If we go back here at the bottom, it will inactivate the making of new glycogen. Okay, so take note of that. Next, we can also see here it activates another enzyme. Okay, this one. And the result here, this enzyme activates glycogen phosphorylase, which breaks down the glycogen to become glucose. Okay? And now the glucose will go to your bloodstream and your cells can use the glucose for energy. So, nana kay source of energy. Diba? Na-break down ang glycogen. So, unsa to ganina ang isa niya ka-resulta? Katong nasa taas. It stops the making of new.
Glycogen.
All right. So hunong ang paghimo sa glycogen. Second, shown here is the breakdown of glycogen into glucose. So you see the two effects are similar, di ba? Stop making glycogen plus break down the glycogen that we have into glucose. Because the goal here is for you to be energized so that you will have glucose in your blood. Okay? So they are related. And then there's another effect, no, of this active protein kinase A. Another effect is it will go to the nucleus. We'll look at this. It will go to the nucleus. It will activate a protein called CREB. And CREB is going to be an activator of a transcription factor, which will make mRNA, which will be translated into an enzyme that is a gluconeogenesis enzyme. What is gluconeogenesis? Gluconeo, bago. Genesis, formation. Unsa man na?
Yeah, synthesis of glucose.
All right. Diba ganina, ang una niya nga effect is stop the making of glycogen. Second, break down the glycogen to make glucose. Third, shown here, is to instruct to make new glucose. Grabe kayang effect, no? The effect is to make glucose. That's the ultimate goal here. Okay? And what was the signaling molecule a while ago? Epinephrine, no? The hormone which gives you energy. stimulates you to have energy, or glucagon, no, the sort of the opposite of insulin. Okay. So, unsa ang second messenger diri, CAMP or inositol triphosphate? Do you remember?
Camp, sir.
CAMP. Kato ganina sa inositol triphosphate, unsa tong ultimate goal niya? Kay hamdo mo? To release what ions?
Calcium ion, sir.
Calcium ions. Okay, for muscle contraction, for example. So, very good. Next receptor is the receptor protein tyrosine kinase. So, kani, lahi na ni siya sa atong G protein coupled receptor a while ago. So, the RPTKs exist as separate transmembrane proteins, and once they are activated by the signal molecule, a ligand, they stick together into a dimer, and they will phosphorylate each other. Look at this, cross-phosphorylation. And once they are phosphorylated, they are able to phosphorylate other proteins, thus activating them. Now, so morning receptor protein tyrosine kinase separate, activated, they will stick together, and they will phosphorylate each other and phosphorylate another molecule. So here are examples. No? So here we have receptor protein tyrosine kinase activated. So there's phosphorylation that happened. And this phosphorylation is used to activate other proteins, like this RAS protein here, kaning RAS. Okay? Kaning RAS, this is important. We will talk about this again later on. Okay? And then, here are other examples. I will not detail on these anymore. Okay? But these are other examples na gina-activate sa receptor protein tyrosine kinase. But we will focus on RAS. Okay. So, let's look at this RAS MAPK pathway. The morning structure sa RAS, kaning ribbon structure. So, activated, kaning pula ang iyang itsura. Inactivated, kaning green. Okay. So, this is our RAS. Now, it's activated by your RPTK. The RAS is a G protein. No, it's a G protein, but it's monomeric. Once, in order to prevent it from getting active, we have this guanine nucleotide dissociation inhibitor. So kaning GDI, gina-prevent niya ang RAS from becoming active. But there is another protein called guanine nucleotide exchange factor, GEF, which causes this to exchange its GDP for GTP. Giilisan og GTP. So, ni-activate na din siya. So, switch on. Ni-active na si RAS. Now, once RAS is active, it can be used to activate another protein, like this target protein here. Okay? And this target protein can be used to activate other proteins. All right. And then, here we have a GTPase activating protein. Okay? Which causes the GTP to become GDP. And once GTP is GDP, the RAS becomes inactive again. So, balik tayo diri sa taas. Ma-inactive na po siya. Okay, so, this is a cycle for RAS. No? Okay. So, however, there is a mutation called RAS gene mutation, which causes the RAS to be unable to be deactivated. So meaning to say, the RAS here is always bound to GTP and it's always active. Now, and this has been associated with cancer. No, kaning RAS nga protein that has mutated and is unable to be switched off. So it can cause, for example, uncontrolled cell division in cells and the formation of tumor and eventually cancer. So this is RAS. Kinsa tong nag-activate kay RAS? Is it the GPCR or the RPTK?
RPTK, sir.
All right. So the RPTK, receptor protein tyrosine kinase. So remember. So this is the pathway for RAS, no? Mao ni ganina. So you have the growth factor. So you have here your receptor protein tyrosine kinase. It will activate RAS. So naa si RAS diri sa ubos, oh. RAS is activated. It will activate RAF, another protein, which will activate MEK, another protein, which will activate ERK, another protein. Okay? So, for this reason, this process or this pathway is called mitogen activating because it promotes mitosis or cell division. No? So, all of these protein kinases are known as MAP or mitogen activating protein. And because RAF, the activation of RAF causes the activation of three more proteins, it's called a MAP triple K, MAP KKK, that's RAF. And MEK can activate two more, so it's called MAP double K. And ERK can activate one more, as we see here at the bottom. This transcription factors, it's called a MAPK. Okay, so, and then the transcription factor will activate the transcription of a gene, which makes a protein that will stop the pathway altogether. So, ang ultimate result ani is to stop the pathway, but the side result is mitosis, not cell division. Now, let's go back to that RAS that cannot be turned off. What will be the result? If RAS here cannot be turned off, your RAF, your MEK, and your ERK will also be going to be turned on again and again and again and again. No? So despite stopping it at this point, it will always turn on this transcription factor. So ipahunong siya pero i-on na pod balik. So mura ka ga-turn off og on og switch sa suga balik-balik. And the result is the cell continues dividing, and again, forming a tumor. So that's why the mutation of RAS is implicated in tumor formation. Okay, and then here, the insulin receptor sort of acts like a receptor protein tyrosine kinase, as you see here. So when it's activated by insulin, so insulin is a signaling molecule, it phosphorylates itself, and it also causes, look at this pathway, the formation of RAS. So RAS gihapon siya associated, no? Okay? And the formation of PI3K, which eventually results to glucose uptake. And a while ago, we talked about RAS, the mutation of which can cause tumors, no? And insulin, which the release of which is caused by an intake of sugar, and glucose will be uptaken by the cell. That's why cancer cells, in order for, if you have cancer cells, no, for example, a person has cancer cells, it is not advised for that person to have a diet that's rich in sugar. Ano man? Because if you eat a diet that's rich in sugar, insulin will be released. The insulin receptor will detect that, and it will cause, look at this, the RAS pathway, cell division. Diba? And it will also cause glucose uptake for the nutrient of your tumor cells. So, it's not advisable to have a sugar-rich diet, especially if you have tumors, no? Kaya makastimulate siya into the proliferation of the tumor cells. So, this is just a list comparing animal cells and plant cells in terms of signaling. So, the first two here, they are the same, but they are different in these last two rows. Okay, so the model for the plant study is the Arabidopsis thaliana. So you can study about that by yourself. So Kani, we will just go back to this calcium ion release. No, mao niya itong gistoryahan ganina. And this is important in muscle contraction. Okay? Because one target of the calcium release is this protein called calmodulin. Okay? And calmodulin, once calcium binds to calmodulin, it becomes activated and it activates a kinase. And that kinase works here. Okay? Remember this pathway? This is the pathway that produces glucose. Okay? So, dili lang di ay for muscle contraction ang calcium ions, but it also activates a pathway that produces glucose for the energy of the muscles. So, you see how amazing that is? Nag-connect ang mga resulta sa atong cell signaling. Okay? In fact, we can also see here the convergence and divergence and crosstalk among signaling. So here on top, you have the G protein coupled receptor. Here at the bottom, you have the tyrosine kinase receptors. Notice, ang ilang epekto is somehow related. Look at this portion, no? Nag-diverge sila. Nag-converge. Okay? And then there are also parts wherein the results diverge, as you see here. And then ultimately here, you see here the part where they crosstalk. Okay? So, nagsumpay ang ilang resulta, even if they started separately and differently. Okay, so, that's a hallmark of cell communication. They can converge, they can diverge, they can crosstalk. Lastly, we have this signaling molecule called nitric oxide. Okay. Nitric oxide is released by your endothelial cell. Ano sa mga endothelial cell? Anyone? Napamantay 7 minutes, so makapangutana pa ko sa inyo. Do you remember what an endothelial cell is? Unsa man nga cross-section? Kung nag-study sa katong nag-microscopy mo sa inyong animal anatomy. This is a nerve ni? Is this muscle? Is this bone? Is this cartilage? What is this? Sa man ang buslot di asto nga, what will pass through that? Wild guess lang. It's okay. Just guess. What do you think this is?
Muscle, sir?
All right. Technically, there are muscles there. But this structure, what is this? Dahil napag-check katapad, oh. Nagyapoy buslot. Unsa man ang sa tunga? Naba yung muscle dahil na buslot sa tunga? Na hollow. Unsa man ni siya? Any guess? Last guess before I tell the answer.
Blood vessel, sir?
Correct. Okay. So, kaning thick layered nga blood vessel, this is the artery, di ba? Familiar mo? Then the thin, kaning lipis ang iyang lining, this is the vein. So magtapad yun na sila perme, di ba? Okay, so this is the endothelial cell, kanang inner lining sa imong blood vessel. No? So the inner lining of the blood vessel, once it receives a signal from a neuron, kaning ACH, acetylcholine, it will create nitric oxide, NO. And the nitric oxide is a gas, and it can diffuse through the endothelial cell into the muscles surrounding the endothelial cell. Mo to imong answer ganina, muscle. So you're technically correct. And unsay resulta sa nitric oxide once it goes into the smooth muscle surrounding the blood vessel? Unsa ni nga word? It will cause the Ang muscle nga nag-tense will relax. No? When the muscle relaxes, the blood vessel will dilate. Kung dako ang agianan sa blood, then blood can rush through into the blood vessel easily. And this mechanism, guys, kaning nitric oxide, is the mechanism that allows for unsa man ang mga body mechanisms nga mag-benefit sa blood flow. No? For example, no, during erection, no? So dili ra man na sa male mahitabo na gihapon na sa babae. So during erection or anything at all no, that requires blood to flow through an organ, this nitric oxide is released so that the blood can easily flow through the blood vessels. So mao na siya ang nitric oxide. So very important, no? So don't disregard nitric oxide, without which it might be impossible to reproduce, di ba? If there's no nitric oxide. So I'll leave this last topic for you. So kamo na lang bahala magbasa ani. This is programmed cell death, no, apoptosis. Let me just talk on the general terms. Caspases. Kaning caspase are the enzymes that are implicated in programmed cell death or apoptosis. So they cause the inactivation of focal adhesions, di ba? So instead ang cell nga maghawid focal adhesion, ma-inactivate, causing the cell to detach from their surroundings. The caspase also cleaves lamins. So ang katong mga nuclear lamina, mga guba, and the nucleus will shrink. It cleaves the cytoskeletal elements, so the cell will lose its shape, and it will activate endonucleases, which are enzymes that will destroy the cell's DNA. So in a way, ang caspase, i-release siya sa cell if the cell is programmed to die, called apoptosis. Ba? So there are two terms here, apoptosis and necroptosis. Ang apoptosis is planned, programmed cell death. While ang necroptosis is namatay ang cell because of an accident. So for example, nabangga ka, nabunog ka. Ang cell dito sa katong nabunog ni mga part, mga matay. And that's necroptosis. That is not planned. But apoptosis is planned. Diba? I think kabalo man taan the formation of our fingers. When we were still embryos, no, naaman tayo web between our fingers, but eventually the webs vanish because the cells in the web die because of apoptosis, programmed cell death. So, this is the signaling pathway, no, of apoptosis, which involves, as you see here, caspases. Okay, the same, but in necroptosis, it involves a different protein which forms pores around the cell, causing the cell to burst and to die also. So there is an intrinsic pathway and an extrinsic pathway for apoptosis, as we see here. So I will not detail on this kay kulang ta sa time, but you can always ask me if you need to ask, no? Message me through eLearn. That ends our lecture on cell signaling. There's a lot to process, no? And maybe for some of you, all of these are new to you. So you really need to read the book in order to understand some more. Okay, so time check, it's already 9 a.m. So are there any questions or concerns? Okay, if there are no questions or concerns, guys, thank you very much, no, for joining in this video teleconference. Our last one will be next week, no, so please stay posted for the schedule. So again, thank you very much. I will post the recording of this soon in our course wall so that you can review what we have discussed. So for now, have a productive day ahead, and you are all dismissed.
Thank you,
Thank you,
sir.
sir.